Hey there, welcome to the video lecture on blood typing. So before getting to the nitty-gritty stuff here, I would recommend that you download and print one of these worksheets that I have available on Canvas. That way you can write in your notes as we follow along. All right, so first thing that I wanted to do is orient you to the chart. So what we have here in the first column is going to be the different kind of blood groups that we're going to be talking about. So blood type A, blood type B, blood type AB, and blood type O. We're going to be looking at their composition of, what do you actually find in the bloodstream with regards to the red blood cells and the antibodies in that person's bloodstream. And of course, we're only going to talk about the antibodies relevant for our blood typing discussion today, but there are a whole lot more antibodies in the blood besides what we're looking at. In the following columns, we'll be looking into what would happen if they received whole blood transfusions from different blood types. And we're going to look at specifically if there was a transfusion from blood type A, blood type O, and blood type AB. So we're gonna omit what happens with blood transfusions from type B blood. And that's because if you understand how it works with type A, then I'm pretty sure you'll figure it out with how type B blood works. Okay, so first word that I want us to look into here is this word antigen. So an antigen gets its name because it is an antibody generator. Antigen, antibody generator. So that's how it gets its name. It's going to be specifically a type of glycoprotein, which is a protein connected to some sort of carbohydrate. And the purpose of antigens is they're going to act kind of like a molecular tag or sort of a identification tag for cells in your body. And all cells in your body have antigens on them. But in this discussion, we are just looking specifically at red blood cell antigens. So, besides this term antigens, let's look at antibodies. So antibodies are going to be what can bind to antigens. Antibodies are going to be these protein structures that are part of the immune system, and they float around in the blood plasma and are essentially just looking to just bind to something that they fit into. So here we have these specific shapes here, like a U shape for these antibodies. So they're gonna fit onto these circular antigens, which we'll look at a little bit later, but as a clue, they're gonna fit onto these guys if they find them. And so, okay, that's just a quick overview on those two words. So let's look at some of these examples. So in type A blood, we have these particular antigens called A antigens. So if you wanted to draw like a little legend or a key, whenever you see a triangle, that's representative of an A antigen. The other antigen that we're going to be spending a lot of time on are the ones that are shaped like circles. And no, not this circle, because that's the red blood cell membrane, but this circle. So those smaller circles are going to be called B antigens. So those are the two main ones we're going to be discussing today. In terms of the antibodies that are floating in the plasma, we're going to also mainly discuss two different kinds of antibodies. The first will be shaped like this, and that will be called anti-A or antibody A. What this means is it is the antibody that binds or recognizes the A antigens. Anti-A antibodies thus kind of attack red blood cells that have A antigens. And the second main antibody we'll look into is anti-B. Those are going to have the more smooth curve to them. And antibody B or anti-B is going to be the antibody that recognizes the B antigen and attacks red blood cells that contain these B antigens. So I'm going to be using this word attack because I think it's helpful for thinking about how this is going to work. So let's start off with this first example of what if a blood type A individual receives whole blood transfusion from somebody with type A. So let's just redraw what kinds of red blood cells are in the donor. So, okay, in the donor, sorry, not in the donor, apologies, in the recipient. So let's draw again the recipient's red blood cell. The recipient has these anti-B antibodies. But then they're going to get a blood transfusion from somebody with type A, which means they are getting a direct sort of injection in a way of red blood cells and also antibodies from the type A individual, since we're doing whole blood. So I'm going to use a red pen to distinguish the donor in red versus the recipient in black. All right, so somebody with type A blood is going to be donating red blood cells with A antigens. And since it's whole blood, the donor is also donating anti-B antibodies. So now that these are mixing together in the same bloodstream, the question here is there going to be any agglutination? Or is there going to be any clumping, is another way to say it. So let's write this. Agglutination, question mark, or clumping? And to answer this question, we have to think about, is anything going to be attacking anything else? And if anything is going to be attacking anything else, we can say, yes, there's going to be agglutination or clumping. But it's so far, well, this is an anti-B, that's an anti-B, but we have no B antigens anywhere. So there's no attacking. So the answer here is no. No agglutination. So this is a safe bet for blood donations. So if you're type A, you can receive blood from a type A individual. All right, so let's try a different thing. So what if an individual with blood type A receives whole blood transfusion from somebody with blood type O? So once again, let's begin with what the recipient has already in their bloodstream. And let's now add what is being what's being IV'd into their body. So somebody who's type O, their red blood cells are going to lack any relevant antigens. You can reference this sheet here, so you can see a type O person doesn't have any relevant antigens for blood typing, but they do carry with them in whole blood. They have anti-B antibodies and they have anti-A antibodies. So let's draw those in. So once again, we can ask this question of whether or not this is safe. Is there going to be agglutination? Will there be any clumping? So it might be tempting to say that no, there wouldn't be clumping. Type O is the universal donor, so it should be safe. But something important to note is we're talking about whole blood transfusions. So we are keeping these antibodies in addition to their red blood cells. So can you find, is there going to be something attacking anything else? Well, you may have spotted it. This anti-A antibody is going to be able to recognize the A antigen and thus bind it and cause agglutination to occur. So clumping does happen, so that is not a safe blood transfusion to do. So how do we explain why people say blood type O is the universal donor? Well, if we were to take away the antibodies before doing the transfusion, then we eliminate any of the worry of anything attacking the donor here. So what they can do is they can centrifugate or spin around the blood, or maybe use other methods too, and they can remove the antibodies from the volume of blood that's being donated. So they take away the antibodies, and thus there's nothing to attack anything else. So while there is agglutination, is there a solution? Is there a way to solve the problem of agglutination? Also, yes. You can solve it by removing the antibodies via centrifugation or maybe other methods. Oh, and something we should also think about here is, what does agglutination even look like when it happens? So I think I'm just going to flip this page over. And so let's think about an individual with blood type A. So they have these A antigens. And then they get some blood donation. Oh, let's also draw on these antibodies. Right? And we are in a blood vessel. Then you get a injection of blood type, let's say we get blood type, let's do blood type O, yeah, since we just did that. So you get an injection of blood type O. When it's whole blood that's injected, then you're going to get the antibodies, the anti-A and the anti-B, and in particular, the anti-A, which is shaped like this, essentially binds the antigens, and then the antibodies themselves, they start to connect with each other. Sort of like this. And when they make bridges like this, they're holding those red blood cells in a tightly packed area, and those red blood cells are no longer freely able to move around. And it creates this ball of red blood cells that you can actually see with your naked eye. But I wonder if we'll see it in the lab. We'll see. But that's agglutination, what it would look like. And by the way, I did omit the other antibodies that would also be there. I omitted these anti-Bs because they aren't really relevant for the agglutination here. All right, so third example here. So what if somebody with blood type A receives whole blood transfusion from a donor with type AB blood? Once again, we can set ourselves up here. Somebody with type AB blood is going to have a red blood cell that has A antigens as well as B antigens. So it's actually a mix of antigens. Type AB blood does not carry any antibodies of what's relevant here. So specifically, they do not carry anti-A and they do not carry anti-B. That should make sense because they would otherwise be attacking themselves, right? They would their own antibodies would then be binding to their antigens and then they would agglutinate. So that would not be possible to survive. So they come with no antibodies. So is there any agglutination that happens in this example? Is anything going to attack anything else? So hopefully you saw that, yes, this anti-B can recognize the B antigen, and so it's actually the recipient is attacking the donor. The recipient is attacking the donor's red blood cells, rather than over here, it was the donor's antibodies that are attacking the recipient. So it's sort of reverse in a way. So agglutination here. Yes, we have agglutination. What about, is there a solution? Is there a way to solve this problem? Well, practically speaking, no. Practically speaking, there's not a way to fix this problem. If you said yes, maybe you said yes because, oh yeah, we can just take away the antibodies of the recipient's blood. But that would mean that you'd have to take all the blood out of the patient, then spin their blood around in a machine, and then remove the antibodies, and then place all 5 liters of blood back into their body. So that is, practically speaking, impossible. So that's why there's not really a solution here. All right, so I'm not actually going to do this row here. I'm going to leave this row for you guys to figure out, because if you understood what happened here, you should be able to figure out the row beneath it. The one that's a little bit different is the third row for blood type AB. So we're going to cover that in this video lecture. So notice blood type AB has the two different major antigens and no antibodies. So in our first example, let's see. So setting ourselves up. So if we have somebody with blood type A donating, we're donating whole blood in this case. Is there agglutination? All right, hopefully you could see that there would be this anti-B that would attack antigen B there. So agglutination, yes, which is bad. Is there a solution for this? Is there a way to fix the problem? Also, yes, there is a way to solve this issue. All that it takes is removing the antibodies from the donor's blood. So you could do this after you've already, after the blood has already been donated, so it's not a big deal for the donor. All right, so moving quickly here, the next example. Blood type AB there. So within a patient's AB blood, no antibodies, but this time we're getting blood from a type O individual. All right, is there agglutination? Definitely yes in this case. Double yes, because we have anti-B that can attack antigen B there, and we have anti-A that can attack antigen A, or A antigens. So doubly agglutination there. Is there a way to fix this? Also yes. This blood type O can donate their blood, and then it can be centrifugated to take out the antibodies. And then here's going to be the last example that we do in this chart. So what if somebody with type AB blood receives from somebody with type AB? We can draw that out if that helps. There's no antibodies though in this scenario, so there's nothing that can actually attack in this scenario at all. So in terms of agglutination, there is no agglutination, so this is a safe transfusion from the start. So a big idea to get from what's been covered so far is, practically speaking, if you have somebody with a specific blood type, let's say blood type A, what is the best option for blood donation for that individual? Should you give that person type A blood, type O, or type AB? So of course, the ones that you want to pick the one that results in no agglutination from the start. That's the easiest solution, because then you don't have to do extra work. Sure, you can give them technically type O, but then you have to fix it up, right? So this is why type O blood, while it's the universal donor, it's only going to be used in emergency situations where there is not enough of the correct blood type to give an individual who's in critical condition. Otherwise, if there is enough type A blood, they will just give a type A transfusion to somebody with type A. That's always the safest bet. Okay, so the very last thing that I want to talk about is this added complexity of the plus and minus. So some of you may know your blood type. Maybe you're A negative or A positive, or maybe you're O negative, O positive, etc. Any of these blood types can be positive or negative. So what that refers to positive or negative, this is going to represent the presence of what's called Rh factor, also known as the D antigen. So this is going to be a third antigen that some people have. In fact, 85% of the population is Rh positive, which means they have this extra antigen. They have the D antigen. For example, we're probably going to do, let's say the D antigen is a square. Let's make the D antigen a square, all right? And for the fun of it, let's make then the D, or the anti-D, this shape, right? So that it matches the antigen. Great, all right. And let's just do one example of this. So let's have a recipient with A positive blood. So we can start off drawing our type A blood, but we can also add on these D antigens. This individual also has the typical anti-B. Does this individual have anti-D? Should they have anti-D in their blood or at all? And so no, they wouldn't have any anti-D in their blood, because otherwise the anti-D would be attacking the patient's own red blood cells. So it would not make sense to have anti-D in the bloodstream. So here's our setup, great. So what if they received blood from somebody who was A negative? So A negative is going to be our donor. We're going to assume that this is a whole blood transfusion again. And we're also going to make another assumption that I'll explain a little bit later, but the assumption is that they've been sensitized to the D antigen. So I'll explain that one a little bit later. But let's draw the donation here. So we have type A blood. Since it's A negative, negative means that's the absence of the D antigen. So we're pretty much done drawing the red blood cell here. Of course, they also have the antibody B. Another thing we're going to assume is that they've been sensitized to the D antigen, so they are going to contain anti-D. So being negative, this is the other 15% of the population here. This other 15% of the population that is Rh negative. And people who are Rh negative may or may not have anti-D. The safest thing to assume is to just make the assumption that the person has anti-D, that it has the antibody that can attack the D antigen. That way, by assuming that, you make sure that your patient is going to be as safe as they can be. So that's why we're making this assumption, just for safety concerns. So, of course, this anti-B is not going to be able to attack anything, anywhere, but this anti-D can bind the D antigen and cause agglutination. All right, so my recommendation is take all this information and work your way through these rows that were not completed, and maybe create your own examples of new examples with plus and minus for the presence of Rh factor. All right, hopefully that was helpful. See you guys later.